Assalamu alaikum. Today I want to talk about chapter 20, electron transport and oxidative phosphorylation. And we are going to cover two sections: the role of electron transport in metabolism and reduction potentials in the electron transport chain. Now, aerobic metabolism is a highly efficient way for an organism to extract energy from nutrients. In eukaryotic cells, all aerobic processes occur in the mitochondria, whereas as we saw in glycolysis, it occurs outside the mitochondria in the cytosol. And until now, we did not see any role for oxygen, although we talk about aerobic oxidation. However, in this chapter, we shall discuss the role of oxygen in metabolism as the final electron acceptor in the electron transport chain. The reactions of the electron transport chain, all of them take place in the inner mitochondrial membrane. So, as I said, in eukaryotic cells, aerobic processes occur in the mitochondria. Glycolysis and anaerobic process occurs outside the mitochondria and the cytosol. Now, the energy released by the oxidation of a nutrient is used by organisms in the form of chemical energy of ATP. Now, the production of ATP in the mitochondria is the result of the so-called oxidative phosphorylation, in which ADP is phosphorylated to ATP. Now, the production of ATP by oxidative phosphorylation is an endergonic process, which means it requires energy. And this is separate from electron transport to oxygen, which is an exergonic process. But the reactions of the electron transport chain are strongly linked to one another and are highly coupled to the synthesis of ATP. How? By phosphorylation of ADP. Now, electron transport chain, series of intermediate carriers that transfer electrons from NADH and FADH2 to O2. The operation of the electron transport chain leads to pumping of protons, creating the so-called pH gradient, also called Proton gradient. Then the reactions of the electron transport chain take place in the inner mitochondrial membrane, and as a result of electron transport, pumping of protons occur to create the so-called proton gradient or pH gradient. Now, this proton gradient represents a stored chemical potential energy and provides the basis of the coupling mechanism. This is also referred to as chemiosmotic coupling. Now, oxidative phosphorylation gives rise to most of ATP production associated with the complete oxidation of glucose. Then, again, production of ATP in the mitochondria is the result of oxidative phosphorylation, in which ADP is phosphorylated to give ATP. And we said this is an endergonic process, which depends on the creation of a pH gradient within the mitochondria as a result of electron transport. Then we explained all that. Oxidative phosphorylation gives rise to most of the ATP production associated with oxidation of glucose. Let's look at this figure here, which shows us the proton gradient is established across the inner mitochondrial membrane as a result of electron transport. Then, if we look down here, at the beginning, electron transport leads to proton pumping across the inner mitochondrial membrane. You see, here we have, this is called the matrix, and pumping of protons from the matrix into the intermembrane space from this side or this side. And as you can see, there is a difference in concentration of H plus. Inside becomes low, so high pH, and in the intermembrane space, high H plus, low pH. This is called a proton gradient. Notice here also, this proton gradient together with the so-called membrane potential, which is a voltage across the membrane, provides the basis of the coupling mechanism that drives ATP synthesis. Now, the complete series of oxidation reduction reactions of the electron transport chain is can be seen in figure 20.2, as I will show you. A point noteworthy to mention is that an average of 2.5 moles of ATP are generated for each mole of NADH that enters the electron transport chain, and 1.5 moles of ATP are produced for each mole of FADH2. Now, the general outline of the process is that NADH passes its electrons into the so-called coenzyme Q in a series. Then CoQ will transfer its electron to a series of proteins called cytochromes, and eventually to oxygen. So again, electron transport carried out by 4 closely related multi-enzyme systems, as we will see, and 2 electron carriers, coenzyme Q and cytochrome C. These are called mobile carriers because they are not fixed. Series of oxidation reduction reactions in which NADH and FADH2 molecules generated in glycolysis and the citric acid cycle, as we saw, you remember, transfer electrons to oxygen. Now, NADH and FADH2 are oxidized to NAD plus and FAD, and these also can be reused. Then oxygen, the ultimate electron acceptor, is reduced to water. Now, results in proton being pumped across the inner membrane into the intermembrane space, creating the so-called pH gradient. Let's look at this figure, which represents a schematic representation of the electron transport chain, showing sites of proton pumping coupled to oxidative phosphorylation. Notice here, at the beginning, NADH transfers its electrons to an FMN. FMN is the flavin coenzyme, flavin mononucleotide, which differs from FAD in not having an adenine nucleotide. Then from FMN to CoQ, coenzyme Q. Then here we have cytochrome B, cytochrome C1, cytochrome C, then cytochrome AA3, and the final electron acceptor is oxygen. Also here, FADH2 transfer its electron directly to CoQ without passing at this point. Now here you have 1, 2, 3. These are sites of proton pumping coupled to ATP production. Now, whenever there is a proton pumping, then there is a ATP production. Now, since FADH2 missed this site for ATP production, this explains the difference in the number of ATP. As I said, NADH, when it's reoxidized, produces 2.5 ATP, whereas FADH2 produces 1.5 ATP. And the difference is here, as you can see from this scheme. Now, let's go and talk about this. Again, these things which I already mentioned, you can read them by yourself. I already talked about these. You can read them by yourself. Now we go to the so-called reduction potentials in the electron transport chain. Now, up until now, most of the energy considerations we have concerned phosphorylation potentials. We saw earlier that the hydrolysis of ATP, the energy released from its hydrolysis will drive endergonic reactions. Now also the opposite is true. When a reaction is highly exergonic, it can drive the formation of ATP. Now, when we look closely at the energy changes in the electron transport, a more useful approach is to consider the change in energy associated with the movement of electrons from one carrier to another. Each carrier in the electron transport chain can be isolated. This can be isolated, this can be isolated and studied. And each can exist in an oxidized and reduced state. مثلا NADH, this is the reduced form. NAD plus is the oxidized form. Okay. Now, if we had two potential electron carriers, such as مثلا NADH and CoQ. Now, the question, are electrons transferred from NADH to CoQ or the other way around? This is determined by measuring the so-called reduction potential. Now, for each of the carriers. Now, reduction potential is a standard voltage that indicates the tendency of a reduction half reaction to take place. Now, a molecule with a high reduction potential, like مثلا CoQ, is paired with a molecule with a lower reduction potential, like مثلا FMN, and this is measured by a simple battery cell, as I will show you. So reduction potential is a useful way to look at electron transport is to consider the change in the free energy associated with the movement of electrons from one carrier to another, as I explained. And this I explained. So this is determined by measuring a reduction potential for the carriers. So if we look at the next figure here, we see that on the right here, in A here, on the right, we have the so-called the reference cell. The reference cell is half cell on the right, where hydrogen ion is in aqueous solution with equilibrium with hydrogen gas. Now, the reduction of hydrogen ion to hydrogen gas, 2H plus 2 electrons to give H2. Now, however, the sample is the one to the left. The sample to be tested is the one to the left. Now, the reduction of hydrogen ion to gas is considered to have a voltage E of 0. Now, the sample is tested in the other half cell. And the electric circuit, as you can see, is completed by a bridge with a salt containing agar gel. Okay, now how can reduction potentials be used to predict the direction of electron transport? Now, in this figure, part A, now shows what happens when ethanol, you see, is oxidized to acetaldehyde. When we put it in this sample half cell, as you can see here, electrons flow away from the sample cell toward H, the reference. This means that the hydrogen ion is being reduced. You remember, in loss of electron is oxidation and gain of electron is reduction. So here H plus is reduced to hydrogen gas. Therefore, the hydrogen H plus pair has a higher reduction potential than the ethanol acetaldehyde pair. That's why electrons flow from low to high reduction potential. Now, if we look at the B here, the reduction of fumarate to succinate here, we put this in the sample. Okay, now here the opposite. You see electrons flow from the reference all the way to the sample. Now here loss of electrons is oxidation, so H2 gas is oxidized to H+. And here we have gain of electron, so fumarate is reduced to succinate. You see? Now, the direction of electron flow and the magnitude of the observed voltage here allow us to make a table as shown in table 21. Yani, here, this is the table. Now, because this is a table of a standard reduction potential, as you can see here, all the reactions are shown as reductions. Reduction, half reaction. Okay. Now, the value being measured is the standard biological voltage of each half reaction, E naught prime. Prime for biological. This value is calculated based on the compounds of the cells, all being at 1 molar, and the pH being 7 at the standard temperature of 25. Now, to interpret the data in this table for the purpose of electron transport, we need to look at the reduction potentials of the electron carriers involved. Now, reaction at the top, if you look here, tends to occur as if it is paired with a reaction that is lower down on the table. For example, we have already seen that the final step of the electron transport chain is the reduction of oxygen to water. This reaction is at the top of this table with a reduction potential of 0.816, a very positive number. Now, if this reaction were paired directly with this one, NAD and NADH, what would happen? Now, the standard reduction potential for NAD plus forming NADH is minus 0.320 voltage. Okay, now its reduction potential is this value, and this is the equation. Now, in order, we want to couple the two reactions. This means that the two half reactions, if we take this and we take the one for NADH, if the two half reactions are paired during a redox reaction, the one for NADH must be reversed. Yani this one, we must reverse this one, NADH plus H plus, to give NAD plus 2H plus 2 electrons. And also, we must reverse the value of E naught, so it becomes positive. Positive plus positive here, you get a total value of 1.136. This is the overall voltage for this reaction. Now, delta G naught prime represents the free energy change of a reaction under standard conditions. Now, the delta G naught prime of a redox reaction is calculated using this equation. You see this equation, where N is the number of moles of electrons transferred, and F is the Faraday's constant. As you can see, delta E naught is the total voltage for the two half reactions. Now, when you expect delta G naught to be negative value, when delta E naught prime is always positive. So, therefore, we can always calculate the direction in which the redox reaction will go under standard conditions by always combining the two half reactions in the way that gives H the largest positive value for delta E naught prime. Now for this example, which I mentioned, delta G naught prime can be calculated to be equal to minus 219 kilojoule per mole. This would be a very large number if NADH reduced oxygen directly. Remember, if we go back to this figure, now this would be a very large number if NADH reduced oxygen directly, as we shall see later. Now NADH passes its electrons along a chain that eventually leads to oxygen, but it does not reduce oxygen directly. It reduces oxygen indirectly by moving from NADH to CoQ to cytochromes and finally to oxygen. Now remember that we have delta G and we have delta G naught. They are different from each other in the same way as delta E naught and delta E are different. Now delta G and delta E refer to the free energy change and the reduction potential under any condition, respectively. But when the components of a reaction are in their standard state, اللي هم ايش؟ 1 atmospheric pressure, 25 degrees centigrade, then all solutes must be at 1 molar concentration. Then we can write delta G naught and delta E naught. Now, the modified standard state for biochemical reactions takes note of the fact that having all solutes at 1 molar concentration includes the hydrogen ion concentration, which means pH is equal to 0, and this is not true. So we define a modified standard state for biochemistry that differs from the usual one only in that pH equals 7. Under these conditions, we write delta G naught prime and delta E naught prime for the standard free energy change and the standard reduction potential. Now, the true direction of electron flow in a redox reaction is also based on the true values of the concentrations for the reactants and products, since the cellular concentrations are never 1